Hi everyone, today's screencast is going to cover treating diseases. Make sure you've got a pen and some paper before we start. When you have got a pen and paper, write down today's LO, which is to understand how vaccines and antibiotics are developed to treat disease. Pause the screencast, make sure you've got this written down. Once you've got the LO written down, pause the video, have a go at these retrieval practice questions from the infection and response topic. Okay, once you've done those questions, you need a different coloured pen, ready to mark your answers. Make sure you pause the screencast and make sure you're doing your corrections if you've got any of them wrong. So as we go through today's screencast, you need to make sure that you are making notes as we go through. They will help you answer the questions later in the screencast. So how can we treat diseases? Traditionally, most medicines or treatments came from natural sources like plants or microorganisms. There are 3 examples here. I want you to make note of these, okay? So the first one, it originates from foxgloves, and it treats irregular heartbeat, so it strengthens the heartbeat, and it's a drug called digitalis. The second one, which most of you will have heard of, is aspirin. It's a painkiller and it originates from the bark of willow trees. And finally, the last one is a drug called penicillin. It's an antibiotic and it originates from mold. Modern medicines and treatments are now synthesized by chemists in labs. So, the most common types of disease are caused by bacteria and viruses. So, we are going to look at how to treat bacterial infections and how to treat viral infections today. This is a quick summary of the information we're going to cover in the next few slides. If you want to pause the screencast and write this down, feel free. It will help you to answer the questions later on. Okay, so this man here is Alexander Fleming. He discovered antibiotics by accident. He was a field doctor treating injured soldiers during World War II, and just after the war, he was searching for a more effective antiseptic to treat wounds. So he worked with bacteria, he grew bacteria, and tried to find different disinfectants that would be able to help the soldiers. So he was away from his lab for a couple of weeks, and when he returned, he found a fungus growing on a culture plate. So a culture plate is what he would grow the bacteria in. The fungus that he found was penicillium mold. He realised that this fungus was producing something that was killing the bacteria that he'd grown. So he tested it on human cells in his lab to make sure that it was safe. This is the kind of thing that Fleming would have seen. These little discs here, you can see that there are patches around the outside where there are no bacteria. So the bacteria is this light grey colour. So we know that these discs must contain an antibiotic. Something is killing that bacteria. Whereas these discs here, you can see they're not antibiotics because they are not killing the surrounding bacteria. But this is the kind of thing Fleming would have seen. So penicillin is the name of the antibiotic and it is released by penicillium mold. Penicillin here, this diagram is showing the action of penicillin as an antibiotic. So it interferes with the building of the bacterial cell wall. So this is your bacteria here. Penicillin acts by interfering with the building of the cell wall. So the cell ruptures and dies when it tries to grow. So there are 2 main types of antibiotics. The first is broad spectrum. These act against a wide range of disease-causing bacteria, so they would be used when there is a bacterial infection suspected, but it's either an infection with a group of bacteria or the group of bacteria is unknown. An example of this is penicillin. The other type of antibiotic are narrow spectrum. These are only effective against a specific group of bacteria. So you would need to know what group of bacteria are causing the infection. if you were going to give this kind of antibiotic to treat a bacterial infection. So we're now going to look at something called antibiotic resistance. If we overuse antibiotics, we can actually speed up the process of bacteria becoming resistant to types of antibiotics. So if you want to take time to read through this diagram, then do so. It's helpful to help you visualize what is actually going on. But usually when you take antibiotics, you are killing the harmful bacteria. Some of those bacteria may have mutated. Mutations happen all the time. Some mutations mean that bacteria is not affected by the antibiotic anymore. So they are resistant to that antibiotic. So the bacteria become resistant. Antibiotic resistant bacteria come about through a process of natural selection. This is where the bacteria that are best adapted to the environment survive and reproduce. So if they are in an environment where there is an antibiotic, those bacteria that are adapted to be able to resist the antibiotic will survive and they will reproduce. As more resistance occurs, it becomes harder to treat. To reduce the development of antibiotic resistant bacteria, it's important that we always finish off courses of antibiotics. This makes sure that all the bacteria are killed and none of them survive to form resistant strains. It's also important that we don't overuse them, so doctors should not prescribe them inappropriately. Antibiotics do not affect viruses. Viral diseases can't be cured by antibiotics because they live inside the host cells. If we think back to the screencast on communicable diseases, when we looked at viral diseases, we know that they live and reproduce inside body cells. The cell membrane, therefore, is protecting them. Viral diseases can be treated with antiviral drugs, but they're difficult to develop because they can damage the host cell while they target the virus. Antiviral drugs usually only slow down viral development, and viruses change their antigens really quickly, and this means that new drugs need to be generated regularly. The best drugs to take are painkillers like paracetamol. These will treat the symptoms of the disease and make you feel less ill. Your task now is to complete the questions below. Try to complete them from memory if you can, then you can use your notes or rewind the screencast if you need help. Pause the screencast, make sure you have a go at all the questions. The number of marks are by the questions, so make sure you've got that number of points in your answer. When you finish the questions, make sure you're ready to mark your answers. And the answers are now on the screen. So pause the screencast and make sure you're correcting any answers that you've got wrong. Okay, we're now going to move on to vaccinations. These are usually administered by needle injections, but they can also be administered by mouth or by aerosol. A vaccine provides immunity towards a pathogen, so it protects you against a disease. Vaccines usually contain a dead or inactive or weakened form of the pathogen. Thinking back to the screencast on the human defences against disease, what do you think will be found on the surface of the pathogen, and what do you think our body will do in response to the pathogen entering our body? Pause the screencast and see if you can write down 2 short answers to those questions. So on the surface of the pathogen, we will find antigens, which are proteins. And in response to the pathogen entering our body, our white blood cells will produce antibodies that will help to target and destroy the pathogen. So once our body has responded to the pathogen that we've been administered in the vaccine, it will have produced the correct antibodies that are specific to that pathogen. So if you were then to become infected with that pathogen, your body would recognise that pathogen quickly, and the production of the antibodies needed to target that pathogen would be sped up. We can see that on this graph here, We can see on the y-axis that we've got the number of measles antibodies that are in the blood, and on the x-axis we've got time. So the measles vaccine is a vaccination with a weakened form of the measles virus, and it results in the production of antibodies and also memory cells. We can see on this graph here that the number of measles antibodies in the blood increases after we have the vaccination. Over time, the number of antibodies does decrease again, but then when we are infected with the pathogen, this is the second time that our body will encounter that pathogen. So it can very, very quickly produce a higher number of measles antibodies, meaning that the measles virus can be fought off very quickly. If we look here at herd immunity, if a vaccinated individual is infected with the pathogen, they can destroy it before they become infectious, so before they are able to pass on that infection to someone else. Consequently, vaccines reduce the likelihood that an individual will spread the pathogen that they have been vaccinated against to other people. If a large number of the population are vaccinated, it's unlikely that someone who's unvaccinated will become infected with the pathogen because it can't be easily passed through the population. This is the principle behind the idea of herd immunity. Now, there are three main scenarios for vaccination. I want you to have a look at the three here, read through them, and then have a look at the diagrams. Now your task is to complete the questions below. Try and complete them from memory and then use your notes or rewind the screencast if you need help. Pause the video and have a go at these questions. Okay, when you're ready, the answers are on the screen. Make sure you're correcting your answers if you've got any of them wrong. Pause the video. So we're now going to move on and look at the development of drugs. New drugs have to be tested for three things: toxicity, are they harmful? Efficacy, do they work? And the dose, so what amount is safe to give someone but is also effective against the disease or illness that you're trying to treat. The results of any testing will be peer reviewed, so they'll be reviewed by different scientists to make sure that the results are described accurately. Any results would then be published in scientific journals for other scientists to be able to read. Preclinical testing is the first stage of the testing. So this is done in a lab and it uses cells, tissues, and live animals. Then we move on to clinical trials. So this will involve healthy human volunteers and patients that are suffering from the illness or disease that you're trying to treat. At first, very low doses of the drug are given to the healthy volunteers. If the drug is then found to be safe, further clinical trials are carried out with the patients to find the optimum dose for that drug. In blind trials, some patients are given a placebo, which is a substance that has no effect on the body, and it allows a doctor to look at the difference in the effect of the drug compared with the effects on the patients that have had a placebo. In double-blind trials, neither the doctor or the patient know who has been given the drug, and this is to reduce researcher bias. Here I've put a summary of the different stages of drug development. You may want to take notes from this. You can use it to help you with the questions that will follow. Your final task for this screencast is to complete the questions below. Try and complete them from memory and then use your notes or rewind the screencast if you need help. The first question is, what are all new drugs tested for? There's 3 marks, so you need 3 different points. And then the last question, the 6 marks available, you need to complete the flow chart below. So where there are blanks, you need to fill in those blanks. And for this last bit here, you need to tell me whether this is a blind or double-blind trial. Pause the screencast, have a go at these, and I'll pop the answers on the screen. Final answers are now on the screen, so make sure you mark through them and correct any that you got wrong.